Very good evening to all of you. Today we will continue with our discussion of previous lecture, that is structure of protein. You can find the lectures of previous part of protein in the YouTube videos. Please let me know after going through the video if you have any doubt. Okay, so let's start our discussion with the structural organization of protein. In our last lecture, we observed that proteins are made up of what? What are the basic units of protein?
Amino acids.
And they are connected to each other via
Peptide.
peptide. So definitely, as proteins are made up of amino acids and they are connected via peptide bond, there is formation of a polypeptide structure. Yes, Rupi.
Ma'am, as carbohydrates are connected with glycosidic linkage and proteins are connected through peptide bonds. Ma'am, fatty acids किससे connected होते हैं?
Ester linkage. They get connected with the glycerol with ester linkage.
Ester linkage.
Yes, I mentioned it last lecture also.
Yes, ma'am, I was a bit confused in the spelling.
Okay, no problem, no problem. So let's understand that this is the basic backbone structure of protein. It's not the functional structure of protein. While teaching you chapter of cell division, I told you people that on the surface of cell there are marker proteins which aptly bind with the specific types of substances. Now, this structural organization of protein and perfection of its binding with a specific type of substance is lying into its three-dimensional structure. Okay, the way you must have observed that for a specific type of lock can be opened only if we are having a specific type of key. Then only we can expect the lock to be open. Similar way, the three-dimensional structure of the protein will decide their functionality. Even if there is a slight change in their three-dimensional structure, the functionality of protein will go forever. Okay, so obviously the amino acid chain produced, which is quite long, need to be folded to get a three-dimensional structure. So in our today's lecture, we will see how the amino acid chain, which is connecting, produced by connecting the peptide bonds with each other, is folding once again in a specific manner. And this folding should not be as much as per its own wish. It should be always following a specific pattern, following a specific thing. Otherwise, if 10 hemoglobin proteins are produced in our body, 10 of them will have different structure. It's not the case. All of them are having exactly same structure. That means there is a specificity in the structural organization. So today we will know that how this structural organization is taking place. So let us understand the structural organization. This in exam, this can come as a long question. So you need to basically draw and describe. So in this, there are 4 levels of organization in case of protein. These are called primary, secondary, tertiary, and quaternary. We will discuss about like one by one. So first level of structural organization is very known to us. It is called the primary structure. So what is this primary structure? Primary structure is nothing but the polypeptide chain we studied, that is having amino acids connected with each other via a peptide bond, and it has N terminal and C terminal. Okay. Now, so if I say you to describe this, you can write this is the amino acids chain is formed by connecting amino acid by peptide bond. So obviously, the bond that is functional over is peptide bond. But can you tell me, is peptide bond a special type of bond, or it is a like we can say that it's a type of a specific type of bond? What type of bond do you think peptide bond is? What are the types of bond you studied, beta?
Ma'am, glycosidic bond, then ester bond and
I'm not talking about biomolecules. You studied what are the types of bond in lower grades, in grade 10.
Ma'am,
Ion
alkenes and
covalent.
alkynes and alkenes.
So peptide bond is basically a covalent bond. Okay, it is a covalent bond. But at it is this type of covalent bond and formation is specially found in case of protein. We call it peptide bond. So that is what you should know, that it's a okay. Now we will go to the next structural organization, which will tell us how this primary structure is folded. So the next structural organization is known as secondary structure. In secondary structure, the peptide bond can coil into a helical structure like this. And this helical structure is called alpha helix. Or it can form pleated sheets. साड़ी के जो पल्लू में प्लीट्स होती है ना, it is somehow similar to that. बचपन में हम सबने फैंस बनाए होंगे पेपर को फोल्ड कर करके. So this is similar to that. फैन बनाए कि नहीं पेपर्स को फोल्ड करके सबने?
yes, ma'am.
So
Yes,
it is
ma'am.
similar to that. So it's known as beta pleated sheet. So simple amino acid chain can fold itself in a way to form either alpha helical structure or beta pleated sheeted structure. Now, what is the description? We can say the peptide chain further coil into alpha helix or beta pleated sheet. So one question that is very natural I am expecting from you. Do you have question? Any question here in this part? Anyone?
Ma'am,
Yes, Mukul.
मैम आपने जो सेकेंडरी स्ट्रक्चर में डिस्क्रिप्शन में लिखा है कि पेप्टाइड चेन फर्दर कॉइल्ड इनटू अल्फा हेलिक्स, मैम उसका मतलब प्राइमरी स्ट्रक्चर फर्दर कॉइल्ड हो जाएगा।
हाँ, प्राइमरी स्ट्रक्चर विल फर्दर कॉइल। इसके अलावा कोई क्वेश्चन नहीं माइंड?
मैम
एनीवन? यस रुकी।
मैम इसमें पेप्टाइड के अलावा और कोई बॉन्ड नहीं लगेगा?
इसमें क्या बॉन्ड लगेगा हमने डिस्कस नहीं किया है। वी आर स्टिल नाउ टॉकिंग अबाउट द डिस्क्रिप्शन। इसमें ऑब्वियसली एक अलग बॉन्ड लगेगा। दिस बॉन्ड इज नोन एज हाइड्रोजन बॉन्ड। You studied about hydrogen bond, how it is formed in chemistry?
No मैम।
No, ma'am.
You studied the concept of polarity in chemistry?
No, ma'am.
No मैम, not yet।
So let me tell you roughly over here what happens. Consider in case of HCl. We know that Cl is quite electronegative, isn't it? Cl is very much electronegative. So what happens is, beta, whenever there is covalent bond formation, we all know that there will be sharing of electron between chlorine and Cl, hydrogen. Isn't it? Yes or no?
Yes
So
मैम।
what do we mean by the word sharing? Sharing matlab it should be equally distributed amongst both of them, है ना? आप और मैं अगर खाना शेयर कर रहे हैं, that means ideally it should be, अगर 4 रोटी है तो 2 मुझे खाने हैं, 2 आपको खाने हैं. But in case of covalent molecules, if one element is very electronegative, another is not, the electron pair is shared. ये शेयर तो होता है, but due to high electronegativity of chlorine, if you see the electron cloud around these two atoms, you can see the hydrogen will be डेल्टा पॉजिटिव, क्योंकि इसकी इलेक्ट्रॉन क्लाउड का एक बड़ा सा हिस्सा क्लोरीन अपने तरफ खींच लेगा. सो सच मॉलिक्यूल्स आर कॉल्ड पोलर मॉलिक्यूल, जिसमें कोवलेंट बॉन्ड होने के बावजूद भी इलेक्ट्रॉन क्लाउड्स का डिस्ट्रीब्यूशन इक्वल नहीं होता है. यस खुशी?
मैम इज देयर एनी रीजन कि कुछ ये चेन होती है वो हेलिक्स शेप होती है और कुछ प्लेटेड, कोई स्पेसिफिक रीजन है क्यों अलग होते हैं?
I will tell you that. But first, you
Okay,
have
ma'am.
any doubt in hydrogen bond? I am explaining now what is hydrogen bond.
नो मैम, नो मैम.
So what happens
Ma'am.
is, yes, beta.
मैम आप ही वापस से रिपीट कीजिए ना।
What to repeat?
मैम जो अभी आपने लिखा है ना वो HCl जो आपने कोवलेंट बॉन्ड शेयर किया मैम, उसके बाद में जो आपने कहा वो।
So H and Cl is connected via covalent bond, which shares that the two electron pairs should be ideally connected with each other. But ऐसा होता नहीं. What happens? क्लोरीन अपनी तरफ इलेक्ट्रॉन को ड्रैग कर लेता है, इसलिए इलेक्ट्रॉन क्लाउड में इनइक्वालिटी दिखती है, एंड सच मॉलिक्यूल्स आर नोन एज पोलर मॉलिक्यूल्स. So now as this hydrogen is feeling itself deprived, if it sees any very electron rich element, it will form an interaction with it. That interaction is known as hydrogen bond. So for hydrogen bonding, one element should be attached with hydrogen, which that element should be electronegative, and it should drag hydrogen's electron slightly towards itself to get a polar molecule. Second condition is hydrogen should find another element which is very rich in electron. So hydrogen will get greedy and find a interaction with it. So that interaction is known as hydrogen bond. So as one of your friend asked me that मैम ये कैसे decide होता है कि कौन सा पार्ट अल्फा हेलिकल होगा, कौन सा पार्ट बीटा प्लीटेड शीट होगा. This will depend on the amino acid sequence of the protein. Why? Because hydrogen bond is possible between two specific amino acids. Consider karo, I have a structure long like this, and I have types of amino acids like this. Now, if hydrogen bond is possible amongst only stars, so the coiling of chain will be somewhat like this, where all the stars will be forming the hydrogen bond amongst themselves, and the other amino acids denoted by the this one, the hearts will remain like this. So depending on the sequence of amino acid, it is decided that hydrogen bond will be created between what type of amino acid. All types of amino acid do not form hydrogen bond with all type of amino acid. So that sequence will decide that it will fold in either alpha helix or beta pleated sheet, because in alpha helix or in beta pleated sheet, these are the parts where hydrogen bonds are possible. So two close parts hydrogen bonds are possible. That's why they fold like this. Is it clear to everyone?
Yes, ma'am.
So
Yes,
now,
ma'am.
now let's talk about the third level of organization, that is called tertiary structure. Tertiary structure is yielded by further folding of the secondary structure. Okay. One very important thing I wish to share here, while you observe the secondary structure, it may happen that in the same polypeptide chain, because polypeptide chains are very long. I'm coming to you, Kandi, one moment. At some part of the polypeptide chain, the chain is folded in the form of alpha helix. In some other part, it is folded in the form of beta pleated sheet. So it's possible that in a single polypeptide chain, some part is alpha helically folded and some part is folded by a beta pleated sheet. Yes, Kanvi, what is your doubt, beta?
मैम अभी आपने बोला कि वो जो हार्ट और स्टार शेप था वो उसी फिगर में ज्वाइन हो गए या कोई भी फिगर ले सकते हैं.
I just gave you example, beta, but they form alpha helix or beta pleated sheet only. They don't form any other structure.
ओके मैम.
So in tertiary structure, it is happening. If you look into the structure of hemoglobin, you will find coiling of proteins like this. Now, if you look into this one protein, you can see that beta pleated sheet, alpha helix, beta pleated sheet, alpha helix, the alpha helix form, and the beta pleated sheet further, again the alpha helix. All of them further coiled to give you this three-dimensional structure. This is what is tertiary structure. So here the alpha helix and beta pleated sheet further coils depending on sequence of amino acid okay and interaction between them. Now, what are the bonds participating like here? Obviously, you can understand from the beginning I am telling that the structure of protein is very specific. If like 10 to 100 proteins are produced in our body, all of them of similar group of protein will have similar structure. Then obviously this folding is not also random. It has to be done by following a specific sequence. So let us understand the bonds participating here is disulfide linkage. So what is this disulfide linkage? You know that in amino acids there are two sulfur containing amino acids, cysteine and methionine. They have sulfur. So the sulfur containing amino acid of one chain can interact with the next sulfur containing amino acid of the same chain to form a disulfide linkage, which is another type of covalent linkage. So this type of linkage can decide the folding of the secondary structure. Second thing that is very important and functional over here is hydrophobic and hydrophilic interaction. So what is this? Have you ever heard this word hydrophobic and hydrophilic?
Yes, ma'am.
So what are they? Can anyone tell me? What is this hydrophobic and philic?
Ma'am, may I?
Yes, Beta.
Ma'am, hydrophobic is the part which stay away from the water, whereas hydrophilic is the part which stays in contact of water.
Which loves water, isn't it? So similarly,
Yes.
in amino acids, some of them are hydrophobic, some of them are hydrophilic, and generally they avoid each other. So hydrophobic ones will avoid the other hydrophobic ones. So while the chain is getting folded, they fold in a way that they can consider the hydrophobic hydrophilic interaction also in mind, so that amino acids which don't want to stay together, they can have maximum distance between them, and amino acids which like each other, they can come close. So that is how tertiary structure is formed. Any questions till this tertiary structure, beta? Anyone? Class?
No, ma'am. No, ma'am.
So the final structural organization, let me once again tell
Ma'am?
you. Yes, beta.
मैम ये टर्शरी स्ट्रक्चर में अल्फा हेलिक्स एंड बी प्लीटेड शीट दोनों का एक ही जैसा स्ट्रक्चर होगा कॉइलिंग का?
नहीं ऑब्वियसली कैसे कॉलिंग खत्म होगा जैसे स्ट्रक्चर में? वो वापस से कॉइल करेंगे ना?
यस मैम.
मतलब कैसे स्ट्रक्चर चेंज हो? They are the coiled in the form of.
मैम मतलब दोनों के डायग्राम आप अलग अलग ड्रॉ करके दिखा सकते हो?
मैम बेटा ये they will be like same only. Even if they are getting folded like this, they will be same only.
ओके मैम.
This is the way they will get folded. Understood?
यस मैम.
Obviously, it will not be same. So now the next important part is the quaternary structure. It's not compulsory for all type of protein. In some cases, quaternary structure may be present, not present. Why? Because quaternary structure is present in case of proteins with more than one polypeptide chain. So the protein which has only single polypeptide chain, for them the final structural organization is tertiary structure. Now, let us start discussing about the quaternary structure. I wish to talk here about the protein called hemoglobin. It has four polypeptide chains present here. Two of them are called alpha, and the other two of them are called beta. So there is like connection between them, and these four functional polypeptide chains are connected with each other to give you the functional quaternary structure of the protein. Okay, so polypeptide chains connect with each other. So what are the interactions or bonds helping in this? These are the same bonds. That is hydrophobic hydrophilic interaction and disulfide linkage. But other than that, hydrogen bond is also functioning in some cases. So that is all about the structural organization of protein. Anyone, any doubt? Even if you have any doubt, we will listen to it later on, because now first let's have a look into the video of this specific part. It's a very nice
We all
video
have things
where
that challenge
things
us.
are
And for
explained
me, it is
very
folding.
nicely.
Sheets, towels, shirts. Let's just
So
say I invest a
you
lot
may
in
let me
anti-wrinkle
know.
underspray. Amazing invention. My
Well,
issue
is
with
the
folding
screen
extends
visible
to
and
paper
sound
too.
audible to
I know
you?
foldables in the classroom can be a powerful way to organize
Yes,
Yes,
concepts.
ma'am.
But for me,
So
it was the
let's
actual
start
folding part that I tended to get stuck on.
from the beginning.
We all have things that challenge us. And for me, it is folding. Sheets, towels, shirts. Let's just say I invest a lot in anti-wrinkle underspray. Amazing invention. My issue with folding extends to paper too.
I
I know
find this
foldables
a
in the
funny
classroom
way
can
to
be a
remember
powerful way
facts.
to organize concepts. But for me, it was the actual folding part that I tended to get stuck on. You may think of folding as a convenience, of a way to take something and make it more organized or condensed so it doesn't have to take up so much space. This is true, but in biology, folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body, just to name a few. We've also mentioned that you are making proteins all the time in a process known as protein synthesis. But the conclusion of producing a long chain of amino acids doesn't necessarily equal a functional protein. There are modifications to a protein that often need to happen in order for it to be functional. By modifications, we can mean many things. It might be adding certain chemical groups, such as phosphorylation, something to definitely explore. But another important event to make a functional protein is, believe it or not, folding. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand. In our cell signaling video, we mention how protein receptors and the signal molecules that bind them can fit together so perfectly to start some type of cellular response. Or in our enzyme video, we talk about how enzymes, which are frequently proteins, have a very specific shape for the substrates that they build up or break down. When we talk about the way proteins are folded, we need to understand the different levels of protein structure, because there are different ways of folding that can happen in the different structural levels. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein. They are held together by peptide bonds. In protein synthesis, amino acids are added to form a
Ma'am,
polypeptide chain.
Yes, beta.
Proteins are made of one or
ma'am,
more of
can
these
you raise the
polypeptide
voice?
chains.
You want me to raise the voice further? Is it like only Rutu not able to hear or others also cannot hear?
Ma'am, the voice is quite low.
Quite.
No, ma'am,
So
it's clear.
I think it must
Genes,
be
which are
a
made of
problem with
DNA,
specific
determine the
person's
order
internet
and
connection
number of these
because
amino acids.
from my
That
point
sequence
it
is
is quite
critical to the
clear.
protein structure and function. In our mutations video, we talk
Is
about
it
how
low for
one amino
everyone?
acid can be changed in
Yes,
sickle
ma'am.
cell
Yes,
disease.
ma'am.
A single
Yes,
change of an
ma'am.
amino acid has the potential to affect a protein's function. We do want to point out each amino acid has a carboxyl group, an amino group, an R group. An R group is also called.
Wait for a moment then. Give me a minute, okay?
My issue with folding,
So
I know
on your request, I'm once again sharing the video. Kindly have a look into it and let me know if you have any doubt.
foldables in the classroom can be a powerful way to organize concepts, but for me, it was the actual folding part that I tended to get stuck on. You may think of folding as a convenience, of a way to take something and make it more organized or condensed so it doesn't have to take up so much space. This is true, but in biology, folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body, just to name a few. We've also mentioned that you are making proteins all the time in a process known as protein synthesis. But the conclusion of producing a long chain of amino acids doesn't necessarily equal a functional protein. There are modifications to a protein that often need to happen in order for it to be functional. By modifications, we can mean many things. It might be adding certain chemical groups, such as phosphorylation, something to definitely explore. But another important event to make a functional protein is, believe it or not, folding. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand. In our cell signaling video, we mentioned how protein receptors and the signal molecules that bind them can fit together so perfectly to start some type of cellular response. Or in our enzyme video, we talk about how enzymes, which are frequently proteins, have a very specific shape for the substrates that they build up or break down. When we talk about the way proteins are folded, we need to understand the different levels of protein structure, because there are different ways of folding that can happen in the different structural levels. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein. They are held together by peptide bonds. In protein synthesis, amino acids are added to form a polypeptide chain, and proteins are made of one or more of these polypeptide chains. Genes, which are made of DNA, determine the order and number of these amino acids. That sequence is critical to the protein's structure and function. In our mutations video, we talk about how one amino acid can be changed in sickle cell disease. Even a single change of an amino acid has the potential to affect a protein's function. We do want to point out each amino acid has a carboxyl group, an amino group, and an R group. An R group is also called a side chain. So even though we have them drawn here like a chain of circles, realize that each of these circles we're drawing is an amino acid like this. Next, we move on to secondary structure. Folding is really going to start to happen. In secondary structure, the sequence of amino acids that we mentioned in primary structure can fold in different ways. The most common ways are the alpha helix and the beta pleated sheet. And which one of those foldings the protein does depends on the amino acid arrangement it has. Both of these shapes are due largely in part to hydrogen bonds. Those hydrogen bonds can occur at specific areas of the protein's amino acids. Specifically, these are hydrogen bonds involving the backbone of the amino acid structure. On to tertiary structure. This is looking at more folding that occurs in the 3D shape of a functional protein. And a lot of this is due to something we haven't mentioned much, the R groups, also called side chains. See, the amino group and the carboxyl group are generally standard parts of an amino acid, although the R group found in amino acids can vary among different amino acids. That means the R group can define the amino acid and can make amino acids behave a certain way. For example, some R groups are hydrophilic. They like water. Some R groups are hydrophobic. They don't. And remember that proteins contain many amino acids, which can contain different R groups, and so different areas of the protein can therefore be impacted based on those R groups. When protein folding is going on, amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups, where are they? They might hang out at the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions. Ionic bonds, van der Waals interactions, disulfide bonds, and hydrogen bonds, all involving the R groups, can influence the folding occurring in tertiary structure. Something to explore. Now, when we've been talking about a protein, we've been talking about a polypeptide chain that has been folded into a functional protein. But proteins can be made of one or more polypeptide chains. And in quaternary structure, you are looking at a protein consisting of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together. Going back to folding, I know what you might be thinking. Who is doing this folding anyway? Are the proteins just folding themselves? Well, the interactions mentioned, like hydrogen bonds and R group interactions, are occurring depending on the protein's own amino acids. One reason why amino acid sequences are very important for protein function. But folding is far more complex than that, and there can be intermediate steps involved when a protein is folding. In fact, there's a phrase you can search called the protein folding problem to learn more about the questions scientists continue to explore regarding protein folding. Research has shown that proteins often have help in the folding process. Chaperonins, for example, are proteins that can help with the folding process. They have almost a barrel shape. Proteins go into them, and the chaperonin tends to have an environment that is ideal for the protein's folding, and this can help the protein be folded correctly so it's functional. Just wish I had something like that for my towels. All of these interactions we mentioned in primary, secondary, tertiary, and quaternary structure are paramount for a mature protein to have its correct shape so it can carry out its function. And that's very relevant. There are many diseases that are related to protein misfoldings. Check out some of our further reading suggestions in the description about that. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, exposed to high heat, for example, you can disrupt the interactions that we had talked about taking place at the different structural levels. This can denature the protein, which disrupts its shape. This prevents it from functioning correctly, and depending on what caused it to be denatured, sometimes you are interfering with many levels of protein structure. Sometimes it's just one or two levels. Sometimes denaturing a protein may be reversible, but in many other cases, it's not. The environment that a protein is in definitely matters for its functioning. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So I guess after this video, no one is having any doubt. It's explained it in a way very nicely, isn't it? Yes or no, class?
Yes,
Yes,
ma'am.
ma'am.
So, yes, beta. Any doubts in protein?
Ma'am,
Yes, beta.
yes, ma'am. Ma'am, can you repeat the disulfide bond?
Disulfide bond. Two amino acids which are having sulfur present in them, there is a creation of a covalent bond which is called disulfide linkage.
Okay, ma'am.
So wherever there is chances of two sulfur containing amino acids to clump close to each other, there is a folding to create due to disulfide linkage.
Okay, ma'am.
Anything else, anyone? It's natural to
Ma'am?
ask doubts. Yes, beta.
मैम वो वीडियो में एक वर्ड यूज किया था चैपर अनियंस, वो क्या होता है?
I'm going to tell you. You know, if you can remember, while teaching you about the cell organelle, I told you endoplasmic reticulum is responsible for synthesis of protein, specifically the rough endoplasmic reticulum. The processing of protein is done by which organelle? Can you remember? I told you. I taught to you.
Nucleolus.
No.
Golgi bodies.
Yes. Golgi bodies is responsible for processing of that protein. So what do we mean by processing? That also I explained that time to you. Can anyone tell me what do we mean by processing? Okay, let me tell you. Processing means proper folding of these amino acid chain into a functional three-dimensional structure. Not only that, some extra parts are added into these proteins through which, or the part which will guide them to specific part of cell. Production of protein is not enough, beta. These proteins need to go to the respective part of cell also. No? They needs to get reached, they should be able to reach to the specific part. How they will reach? एक बार Golgi bodies से वो निकल गए, how they know where they should go? So the guiding molecules are there. Now, what are chaperones? So chaperones are, in other terms, called mother fairy of the proteins. Why? They not only guide proteins for folding, they kind of hold them, and obviously without chaperone also protein chain can fold. It's not that it cannot, but it will take too much time. So these chaperone proteins are guiding the amino acid chain that how to fold, so that folding can be done comparatively faster. Second thing is they mentioned that proteins of specific organisms are specific type. For example, if any organism is habituated to live in cold climate or habituated to live in very hot climate, when we will learn classification, you will be able to know there are bacteria which can live in temperature more than 100 degrees centigrade. How they do that? आपके हाथ में अगर गर्म पानी गिर जाए, it will get destroyed. But they prefer that temperature only. More than 100 degree temperature is very, very comfortable temperature for them, and they prefer to stay like that over there. Okay. So that means their body is having certain kind of special mechanism which help them to tolerate the this condition, isn't it? These proteins are called heat shock protein. These protein give them ability to survive in heat shock condition, that is very low temperature or very high temperature. And depending on the temperature, or I can say the kind of resistance it is getting for environment, the structure of protein obviously change. The structure of proteins of marine animal and freshwater animal will be of different structures. Although they may have similar function, but their structures will be different. So these are the diversities which are there in this protein structure depending on organism to organism. Any other question, anyone?
No, ma'am.
Sure. So now we will discuss today the last part of this chapter, that is called the shapes of protein. See, if you look into shape of protein, they can be of various shapes, but basically two like types of protein that is observed. One is fibrous type and the other one is globular type. So what do we mean by this? The structure of hemoglobin displayed over there was the type of protein which was globular. That means the amino acid chain fold in a way to give you the structure which is globular in a structure. That means it has a globose shape. Fibrous proteins, for example, like keratin protein, which is present in our hair, do not have that globular structure. And due to change in these two structures, they have more like a elongated fiber-like structure than to get folded in a compact globular form. So as a result, fibrous proteins are generally insoluble in water, whereas globular proteins can make them very well adapted with the water to form either solution or colloidal solution. Okay, so this is all about the types of protein. Next thing that I wish to share here is about events that takes place. There are words called denaturation and renaturation. So what do we mean by this? For denaturation and renaturation. See, we know that the folding of protein chain is not having bonds which are very strong. For example, hydrogen bond is not that much strong. The hydrophilic hydrophobic interaction is not that much strong. Although disulfide link is comparatively stronger, the van der Waals forces that is also operational is not that much strong. So, in such situation, if you are applying heat to the protein, the like structuralized organization of protein will get destroyed. Means, consider a situation you have a coiled form of protein. Once you apply heat, it will be existing in comparatively less folded structure. Okay, this phenomenon where there is uncoiling of protein, we call this phenomenon as denaturation. Okay, it is observed if there is minor change in the temperature. In that case, if the temperature is reverted back, for example, there was let's say 5 to 10 degrees centigrade increase in temperature. In cases, depends on protein to protein. Once you get the temperature back into the optimum zone of protein, again an opposite procedure takes place, that is called renaturation. Regaining of three-D structure of upon reaching optimum temperature. So now the question is, if proteins have this ability to self-heat, आप उसको टेम्परेचर दे दो, वो अनकॉइल हो जाएगा, जैसे टेम्परेचर वापस आएगा वो कॉइल हो जाएगा. So denaturation and renaturation goes hand in hand. Why then there is destruction of protein structure? For example, if you are boiling an egg, I know that many of you are vegetarian and never ever observed boiling of egg. But if you would have been seeing, you can see that the liquid part of egg is converted into a solid part, and its structure is changed. So do you think that it's always possible to reverse the uncoiled structure into coiled one? No. It is only possible to a narrow range of temperature. Once you are crossing that limit, protein will go for degradation. That is not denaturation, we call it degradation. So if temperature change is extreme, renaturation is not possible. As peptide bond breaks down. So आपकी जो बेसिक स्ट्रक्चर है, जो जिसने अमाइनो एसिड चेन को जोड़ के रखा है, अगर वही टूट जाएगा फिर तो पूरी अमाइनो एसिड की सीक्वेंस उल्टी पुल्टी हो जाएगी. Then how can it get renatured? So at very high temperature, protein is getting degraded. And if there is minor change in temperature, it can have that ability to self-heal. It can come back, denaturation. It can get denatured and come back to the original state by renaturation. Understood this concept of denaturation and renaturation, beta?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So now
Last thing
we
we
will.
haven't mentioned. Each protein has one last thing we haven't mentioned. Each protein has many
We will like have now the like look into the modification of protein. So I am going to share what type of modification actually proteins are. This part will be more understandable to you in higher grades, but I thought I should share to you. It will be easy for you to understand the condition. So the part I am sharing now, you don't have to remember it. I am just sharing you for your knowledge, or I can say better understanding of the part. Is the screen visible and sound audible?
After a polypeptide chain is formed, it's going to be folded into its secondary and tertiary structure.
Screen visible and sound audible, no?
Yes,
Okay.
ma'am.
Into a very specific 3D conformation or shape. And at this point, we can start calling it a protein. But this protein may not be ready to carry out its function just yet. There might be some additional protein modifications that need to be made to this protein before it can be functional. And those are called protein modifications. There are two different types of protein modifications. The first type is co-translational modification. And that means that these are modifications or changes that happen to the protein, or actually to the polypeptide, while it's being translated. So let's say we have the ribosome right here, and we have a polypeptide that's being formed. So these changes are going to happen while the polypeptide is being formed. And an example of a co-translational modification is acetylation. And what happens during acetylation is the first amino acid in the polypeptide, which is usually methionine, is going to be removed. And in its place, we put an acetyl group. So let's just draw an acetyl group. And acetylation happens to 80 to 90% of eukaryotic proteins. But the significance of this modification is not known very well. We're actually trying to figure out what the purpose of this modification is. The other type of protein modification that happens is post-translational modification. And actually, most protein modifications fall into this category, and the examples we're going to discuss in this video are all post-translational modifications, and those modifications happen after translation. Many post-translational modifications happen in the endoplasmic reticulum and the Golgi apparatus, but not all of them. So let's go through some examples. So the first post-translational modification I want to talk about is glycosylation. And glycosylation, well, you can look at the word, the prefix glyco tells us that it has something to do with a carbohydrate. And so glycosylation is the adding of a carbohydrate to a protein. And most of the proteins in this video are all in green. And glycosylation usually happens to proteins that end up being embedded in the cell membrane. So you can see here we have a cell membrane and this protein embedded in it, and then there are these carbohydrate groups attached. So here's a carbohydrate group attached, and here's another one. And glycosylation helps to identify different types of cells. And one very common example of where we use glycosylation is in the ABO blood groups. So let's take 4 different red blood cells, and let's just say that each one of these red blood cells belongs to a different person. And so red blood cells have these proteins embedded in their surface. And these proteins are going to have many times different carbohydrate groups attached to them. So let's say that this person right here has this particular carbohydrate group attached to it. That makes him blood type A. Let's say that this person has a different type of carbohydrate attached to the protein. Let's say it looks something like that. That makes them blood type B. Let's say that this person has actually both of those carbohydrates attached to his red blood cells. That would make him blood type AB. And let's say that this last person does not have any carbohydrates of this category attached to the proteins on his red blood cells, and so that makes him blood type O. So here's a very common example of how glycosylation is used in the identification of different types of cells. Let's go on to a different type of post-translational modification that's pretty similar, and that is lipidation. And lipidation is when we add a lipid to a protein, also a protein that's going to be attached to the cell membrane. And this lipid we're looking at is actually an example of a GPI anchor. And GPI anchors are lipids that help to attach or tether proteins to the cell membrane. And just to give you an idea of maybe why this would be necessary, so to quickly review the structure of the cell membrane, we have these hydrophilic heads. That means that they are polar. And then we have inside these hydrophobic tails. And that means that they are nonpolar. And so the protein has both polar and nonpolar parts on it, and maybe it just doesn't attach well to the hydrophilic portion of the cell membrane. And so this GPI anchor, a lipid, kind of plunges into the lipid or hydrophobic part of the cell membrane. And we know that substances that are similar, like substances that are both hydrophobic, attach very well to each other. So this lipid, which is hydrophobic, attaches very well to the inner part of the cell membrane that is also hydrophobic. And so that's how it helps to attach the protein to the cell membrane. And both glycosylation and lipidation usually do occur in the endoplasmic reticulum or in the Golgi apparatus. Let's move on to some protein modifications that have more to do with the activity or the function of the enzyme and less with the structure. So one very, very common protein modification I want to discuss is phosphorylation. And phosphorylation is basically the adding of a phosphate group to a protein or to an enzyme. And then phosphorylation comes along with dephosphorylation is when you remove a phosphate group from the enzyme or the protein. And I'm just going to make a little bit of room over here. And so what you're looking at is this schematic diagram of the sodium-potassium pump that's found in basically every animal cell. So the sodium-potassium pump. And another name for the sodium-potassium pump, which is the enzyme that you're looking at, is the NAKATPase. And so this enzyme or protein, the sodium-potassium pump, is responsible for maintaining the proper osmolarity of sodium ions and potassium ions in and out of the cell. And so let's see how phosphorylation regulates this protein. So, and again, the proteins that you're looking at, they really all represent one protein. It's just we're going through the motions of the changes that happen to this one protein. But it's not like we have it's not like we're looking at 6 different proteins in the membrane. So here's our first step. And when you look at this enzyme, you can see that there are 3 receptor sites for a particular ion. This is the sodium ion, represented by these dark blue circles. And then there are 2 receptor sites that look kind of more squarish, and those are receptor sites for the potassium ions, and those are represented by these light blue squares. So those are the potassium ions. And so back to the first step in what's happening here. The sodium ions will attach to the receptor sites on the enzyme. And just to clarify, this is the intracellular space. So this is basically the cytoplasm. And so these sodium ions are coming from the cytoplasm, and then out here is the extracellular space, the outside of the cell. Anyway, back to our first step. So the sodium ions attach to the receptor site, and when the receptor sites are full, it's going to cause something to happen. It's going to cause an ATP molecule, adenosine triphosphate, to break down into ADP, adenosine diphosphate plus phosphate. And then this phosphate will attach itself to the protein. And that is phosphorylation. And so when this protein gets phosphorylated, when the phosphate group is attached to it, it causes there to be some sort of change in the conformation of the protein. And that change in conformation causes the protein to turn itself around by 180 degrees and face the outside of the cell. And that's what you're looking at right over here. So our protein is still phosphorylated. I know I drew it in a different place, but just, keep in mind that there's still a phosphate group attached to that protein. And then the protein releases these sodium ions into the extracellular space. So the next step is number 4, and again, we are still phosphorylated. There's still a phosphate group attached to our protein. And so in the next step, the potassium ions on the outside of the cells will attach themselves to the protein on the receptor sites. And take note, there are 3 receptor sites for sodium, but only 2 for potassium. When the potassium receptor sites are full, it's going to cause a different change to happen. This phosphate group is going to be removed, and that is called dephosphorylation. So that is dephosphorylation. And so that phosphate group ends up in the inside of the cell and it gets recycled in some other way. And when this protein is dephosphorylated, when that protein is removed, it's going to cause a different change in the conformation of the protein. And that change in conformation causes the protein now to turn around again by 180 degrees, and it faces the inside of the cell. So that's our 5th step. You can see the protein is facing the inside of the cell. And in the last step, the potassium ions are released into the inside part of the cell, and then we're back to our first step. And so you can see that the phosphorylation and dephosphorylation basically regulates the activity of this protein. And the end result that we're trying to get to is that we want there to be on the outside of the cell a rather high concentration of sodium, and a rather low concentration of potassium. And on the inside of the cell, we want there to be a rather low concentration of sodium and a relatively high concentration of potassium. And that's accomplished by the fact that for every 3 sodiums that are pumped out, 2 potassiums are pumped in. And so here's an example, a very common example of how phosphorylation regulates a protein. And this doesn't happen just in the sodium-potassium pump. It happens in many, many enzymes and proteins in our bodies and in cells. Let's move on to 2 other protein modifications that also have to do with regulating an enzyme or something similar to that. So the next protein modification I want to talk about is methylation. And in particular, the methylation of certain proteins called histones. And those are these green circles that you're looking at. And histones are these proteins around which DNA wraps itself. So they're found in the chromosomes and they help to package DNA in a very tight and organized manner. And sometimes histones are methylated. So let's put some methyl groups on our histones. And methylating and demethylating histones helps to turn certain genes on and off. And so here's another example of a protein modification helps to regulate activity, but in this case we're regulating the activity of genes as opposed to proteins. And another protein modification I want to bring up is proteolysis. And by looking at this word, proteo means protein and lysis means to break something down or to cut something. And proteolysis is sometimes to take a protein and activate it, we need to cut it. And in fact, the insulin has to be cut twice before it's activated. So let's cut this protein twice. And you may have actually heard of the term zymogen. And a zymogen is an inactive form of an enzyme. And sometimes the way to activate a zymogen is by cutting it. And that's proteolysis. There is one more protein modification I want to discuss. And that is sometimes we add a protein ubiquitin, well, to another protein. So that protein I just added is ubiquitin. And this process is called ubiquitination. And what ubiquitination does is it basically marks this green protein for degradation or for breakdown. So within a short while of being ubiquitinated, this protein is going to be destroyed and the different parts are going to be recycled. Let's just quickly review the post-translational modifications that we discussed. So we talked about glycosylation and lipidation. And those are two protein modifications that generally happen to proteins that end up being embedded in the cell membrane. So glycosylation was the adding of a carbohydrate group, which helps to identify certain cells, and lipidation is when you add a lipid to a protein that generally helps to anchor a protein to the cell membrane. Then we talked about phosphorylation, methylation, and proteolysis. And these all had to do with activating or deactivating an enzyme or genes. So phosphorylation was we brought the example of the sodium-potassium pump, which is basically regulated with phosphorylation and dephosphorylation. Then we spoke about methylation, which can basically helps to turn on or turn off certain genes. And proteolysis is the way in which many enzymes are activated. And the last post-translational modification we talked about was ubiquitination. And ubiquitination marks a protein for degradation, and then the various parts are recycled.
So this part is interesting. I guess you will be able to know that only the basic structure is not all right. For example, I just want to ask you a simple question. They talked about proteolysis beta. Can you give me,
hopefully by now you're familiar
can you
with the
give me
central dogma
example
molecular
of
biology
like
that tells us
a process where from an inactive enzyme is getting activated to go for any process? Have you studied any in 10th grade? जिसमें inactive enzymes था and it was activated due to proteolysis. Anyone? Okay. Have you ever wondered that stomach is made up of what? Cells only, no? I guess I am audible to all of you.
Yes, ma'am.
Cells have protein present in them, isn't it? Yes or no?
Yes, ma'am.
So why like the wall of stomach is not getting digested? क्यों वो खुद digested नहीं हो जाता? Because stomach is having proteolytic enzymes through which stomach itself can digest its wall, no? But ऐसा होता नहीं है, क्यों नहीं होता? Have you ever wondered? Anyone?
म्यूकस की वजह से.
Not actually correct. Mucus is there.
मैम
Yes. Yes, beta.
मैम एंजाइम्स और एसिड्स के कारण.
Acids के कारण, enzyme के कारण means I didn't get. Do one thing. Tomorrow, come up with an answer. I just want to know what you search and you want to come up.
Ma'am, can you just repeat the question?
Although the wall of stomach is made up of proteins only, because stomach means they will have cells are having lot many proteins present in them. Why these proteins present is not getting digested by the protein digesting enzyme secreted by stomach? They should be digested, no? Why it is not? So I am concluding our lecture over here today. Tomorrow try and find out. No problem if you don't find. We will discuss it tomorrow. Okay? Yes, Ayushi.
मैम मतलब इस वीडियो में कई सारे टर्म्स थे जो समझ में नहीं है, बहुत सारे नए टर्म्स थे.
Actually, it's not understood by you because organic chemistry में आप वहां तक नहीं पहुंचे हैं. जैसे acetylation means addition of acetyl group, which is COCH3 group is called acetyl. Methylation means addition of methyl groups, that is CH3 group को methyl group बोलते हैं. Then glycosylation and lipozization is, I guess, understood by you. That is simply by addition of protein, carbohydrate and lipid. Then
Yes,
there was
ma'am.
one term called ubiquitization. उसमें ubiquinone नाम की एक substance होती है that is added to protein.
मैम
Yes, बेटा.
मैम डीफॉस्फोरिलेशन समझ में नहीं आया उस वीडियो में.
Phosphorylation means addition of phosphate. Dephosphorylation means removal of phosphate group.
और मैम zymogen क्या था उसमें?
हाँ, तो ढूंढो, आंसर ढूंढो. It's related to the question I asked you. Try and find out.
Okay, ma'am.
Okay, so we will say bye to each other from here. We will meet once again tomorrow. Let me see what answers you found out of my question. Okay? Okay, beta.
Okay, ma'am.
Bye to all of you.